Don't know, I'm Holly Michael, and I'm co-PI and research lead on Project WiCCED. I'm really happy to introduce Tom Hanson. Tom has a bachelor's in bacteriology from the University of Wisconsin-Madison and a PhD in microbiology from UC Davis, as well as a postdoc at Ohio State. He is currently a professor in the School of Marine Science and Policy at UD with a joint appointment in biological sciences, and he is the interim associate director of DBI, the Delaware Biotechnology Institute. He has done a number of great things, but I'll mention some of the teaching related things that he's been up to. So he was instrumental in launching the Interdisciplinary Microbiology graduate program with Unidel funding. He's teaching courses in graduate microbiology, bioinformatics, and undergraduate marine science courses. And he's a trainer in the chemistry biology interface graduate program. So Tom's leading our WiCCED Microbiome Core, which is one of 3 core groups that interact and support research across this really big project. So those of you who are involved in Project WiCCED, if you're not already working with Tom, please think about how you might engage with that core for your research, because we'd really love to have it, expand into all pieces of the project. Tom is a pretty amazing bread baker. We were just talking about that, and also a pretty wicked soccer player. We used to play together before we retired due to COVID and old age. But he, professionally, he works primarily on sulfur metabolism and also a range of other things. And so we'll hear about some of that today. So Tom, thank you for joining us, and we look forward to your talk.
Great, thank you so much for giving me the opportunity here to do this. And first off, what I'm gonna do is actually introduce the person who I am lucky enough to be responsible for all of this. She actually makes everything that I'm going to talk about possible today, and that's my graduate student, my PhD student, Alexa Bennett, who's actually supported by Project WiCCED funding through the Microbiome Core, and all the data that I'm going to talk about today is data that she's generated and that we've analyzed together. So she's going to give an overview of what she wants to do with sort of the science end of that. And then I'll talk a lot about sort of the background about how we do it and why we've made certain choices that we have and what we can conceivably do for any of your projects. So Alexa, why don't you go ahead and unmute, and then I will change slides for you, like I said.
Okay. So today's discussion will be on the ambitions of the Delaware Microbiome Project. Next slide, please. Okay, a little bit about the project itself. In an attempt to characterize the Delaware microbiome, we are crowdsourcing samples to achieve broad coverage of Delaware's water, sediment, and soils. The talk for today will further discuss the data analysis, specifically as it pertains to the Delaware Microbiome Project, which falls under the Microbiome Core. Next slide, please. Okay, so our aim is to determine regional microbiome patterns, and then from those formulate predictions. The data which is being generated is microbial relative frequency, abundance, and diversity using taxonomic markers for both prokaryotic and eukaryotic organisms in comparison with functional gene markers to capture 5 different nutrient cycles. Each sample we are saving metadata for, which we have the location, the date and temperature of medium at time of sampling, a water or soil chemistry panel, and then we can also generate additional metadata by proxy. So an example of this would be if we have the location and we have the Delaware soil map, we can determine the soil type. For example, is it a sandy loam? So from this, we'll be using the various data points to observe trends and correlations. Next, please. Okay, so the pie in the sky, as Tom likes to say, aspiration is for us to use machine learning. So this is in comparison with the data core under Project WiCCED to formulate predictions about the microbial community from only 2 photos. We're taking one at the horizon for each sample and one directly down on the sample. Next slide. Okay, with that, I'd like to thank my funding, Project WiCCED, Dr. Hanson, the data core lab members, both previous and current, as well as my graduate committee. Final slide. So questions, comments, and hopefully for a 2-minute talk, there are no concerns, but whatever you have.
All right, if there's nothing there, also please feel free to put them in the chat, and maybe Holly can keep track of that, or someone can, and you know, read them out at the end as we go. So thank you, Alexa. I'm going to talk again about all of the stuff that she's been doing.
Tom?
Yeah.
Do you, I'll monitor the chat. Do you want us to interrupt you if there are sort of clarity?
It does, yeah, that's totally fine. I'm good with tangents and interruptions, so.
Okay, sounds good.
All right. So I figured that you know, this is Project WiCCED. It's a big research infrastructure improvement project. I should have a fairly formal sounding title, which is who are Delaware's microbes and what are they doing? First insights from improved research infrastructure. And again, I'd like to thank the team for including me on the project. And then Project WiCCED itself is funded primarily by the National Science Foundation, with a required matching component from the state of Delaware, and is maintained and administered by the Delaware EPSCoR office. But I really can't resist poking fun and hopefully repurposing statements from politicians that may have slightly checkered pasts. Any rate, I had this for a talk that I gave about 2.5 weeks ago, which is Project WiCCED's Microbiome Core, we're here to help. And it's based on this quote from Ronald Reagan, which is the 9 most terrifying words in the English language are, I'm from the government and I'm here to help. We are a government supported entity, but I'd like to convince you that we actually can help, and it is beneficial to interact with us. So here's an outline of what I'm going to be talking about today, a brief introduction and reintroduction to what the Microbiome Core is. I'll talk a little bit about why we want to care about microbiomes and how we've decided is sort of the best way to approach analyzing them, with a focus on reproducibility and quality for very little cost. I'd like to hopefully convince you in answering the third question that yes, indeed, this does actually work. Give you a couple of vignettes of things that we can say to date in the project, and then also give you a little bit of a picture of where we're going from here. So first up, what are we? What is this idea of the Microbiome Core? As Holly said, the Microbiome Core is one of the enabling cores of the, sorry, let me get laser pointer here, of the of Project WiCCED. And I must have had something to do with this because I put us right in the center, because clearly we are incredibly important to the success of everything. Yeah, no, it's just how it worked out. What we're really striving to do is provide information to both threat assessments to help analyze sites and samples that researchers in the project have, that ultimately lead to solution development, and at the same time actually contribute to the education outreach mission of this and the diversity mission of this project, to ultimately enable decision support tools, early warning systems. And in fact, I'll talk a little bit about a site that we have that we've analyzed where I think we've got a strong case that, yeah, something funky has gone on at this particular site and we need to take a closer look at it. So I'm not going to go into too much detail because this is a Project WiCCED seminar, and most of the people that I've seen on the call so far are relatively involved. If you've got questions about how Project WiCCED works overall and what this sort of house-like structure is, please direct them to Holly or Amy Slocum, or go to the Project WiCCED homepage and we can address your questions on that there. Who we are is really, I'm co-lead with John McDowell at Delaware Technical and Community College, and we also have partners in the Data Core, Tina Callahan, Dan Leathers, in bioinformatics, Sean Polson at Dell State with Cal Calvacharla, and Kevin Schumann at Wesley, who I actually saw was on the call as well. And in fact, some of the samples that we'll be talking about are samples that were collected by Kevin to get data that's ultimately going to be used in the classroom. So our transformative goal here is to provide rapid, low-cost access to microbiome data for this proposal and to actually establish this core to survive beyond the length of this proposal and become established and provide this service on a continuing basis at the University of Delaware as a lasting improvement to our resource infrastructure. So what we want to do is implement microbial community profiling and genome sequencing computation pipelines, and not only computation pipelines, but also sample analysis. Basically, all you should have to do, our mission is that as a researcher or as a classroom, all you should have to do is give us a sample and some information about that sample, and then we can take care of everything in the middle before giving you some data that you can feel confident in. The way that I phrase it is that we take care of making the sausage and we follow all the FDA regulations for making sure that that sausage is nutritious. And we also care about it, so we make sure that the sausage is tasty. And I'll come back and talk about that a little bit at the end. I'll talk about the Delaware Microbiome Project. I'll mention briefly the Delaware Community Sequencing Program. And then at the end, I'll provide a link to a page, which then has more information about those. And ultimately, the science that we want to do is what Alexa was talking about, meta-analysis of data that we collect through these efforts to really try to get to predictive understanding of microbial community structure and function in the context of environmental data that we can, in many cases, collect or obtain more quickly and more cheaply than the microbiome data. So, what are microbiomes? Why should we care about them? And how are we actually going about providing this service? So a microbiome is really the collection of all microbes or the microbial community that occurs in an environment. Microbiomes are dynamic things. When I talk about a microbiome profile or a microbial community profile, what I'm talking about is really a snapshot of the community at a point in time. So microbiomes can vary through time, they can vary through space, and they can vary as a result of long-term environmental trends. And those are all some of the things that we're trying to tease out in this. Microbiomes consist of bacteria, archaea, eukaryotic microbes like algae, fungi, protists, and viruses. For the purposes of our core, we are going to ignore viruses, not because they're not important, but because, well, realistically, they're hard. As I'm going to talk about, we have marker genes that we know exist in all cellular life forms. And that gives us a really good handle to be able to get information from the environment about who was there. Unfortunately, for viruses, there is no universally conserved marker gene. And so simply put, the techniques that we have in our toolbox within the core are really not going to help us out in understanding viruses. We can help you plan if this is something that you're interested in, but there are other groups at the University of Delaware, namely groups of Sean Polson and Eric Womack, who focus exclusively on viral metagenomics, and they would be the people to talk to about that. So what about why? Why should we care about microbiomes? Well, this is an image of a single colony of a cyanobacterium called Trichodesmium thibautii. This is microbiology that you can do with forceps. These colonies are big enough that you can actually pick them out of ocean water samples. And what these organisms do in the open ocean is they take water, carbon dioxide, and nitrogen gas from the atmosphere around us, and they convert that into oxygen gas that we require for our own continued existence on the planet. They convert it into biomass that supports the entire marine food web that occurs underneath them. And importantly, they provide ammonium that also supports that marine ecosystem food web. Not only is this microbiology that you can do with forceps, but this is microbiology that you can do using satellites. This is a shot of a trichodesmium bloom off of the northwest coast of Australia, and this is about 250 kilometers on a side. The bright white areas here are clouds, but everything in the middle here is a patch of trichodesmium colonies that are actively producing oxygen, biomass, and ammonium in this relatively infertile part of the ocean and driving the entire marine food web in this section of the ocean. So microbiomes are key to all of these things that we sort of collectively call biogeochemistry. It's microbial biomass and microbial enzymes that drive these key transformations. Now, the problem is that microbiomes are complex. So a microbiome may contain hundreds or thousands of different microbes. So how do we affordably and in some way not overwhelm ourselves with too much information about what's going on in a system so that we can make decisions about which systems we may want to take really deep dives into and which systems we don't need to know much more beyond sort of a surface level understanding. As I've mentioned, microbiomes maintain the biosphere. I mentioned this reaction in the context of Trichodesmium thiebaudii, but that's just one step in the biogeochemical cycle of nitrogen. Now, as we're all aware, and what's a big component of Project WiCCED is that humans are actually influencing biogeochemical cycles. In fact, for this particular reaction, the fixation of nitrogen gas into ammonium, human populations have doubled the amount of material that's fluxing through this reaction by the introduction of a purely chemical process called the Haber-Bosch process for fertilization of agriculture. This was a large part of the Green Revolution that's helping feed an expanding population on our planet, but it also has consequences. We apply that fertilizer as ammonium nitrate to fields, and those are both very mobile species in groundwater. They end up in surface waters as organic nitrogen in many cases, and they drive cyanobacterial blooms. And in fact, we'll talk about a focus project that we have that's a component of our own undergraduate research in my lab / the core that's really looking at this across Delaware. Now, these key reactions in green here are ones that are predominantly driven by microorganisms. And for each one of these arrows, we have characteristic enzymes or characteristic genes that encode enzymes that carry out the different steps in this project or in this cycle. So the gene here is called NIF, which is short for nitrogen fixation. That produces an enzyme that catalyzes this transformation in a given microbe. Because this is a gene, we can use tools like PCR to amplify functional genes in addition to genes that tell us about the phylogenetic or taxonomic identity of the organisms that are present in a community. So we can assess whether or not genes for each step within the biogeochemical nitrogen cycle are present, how diverse they are, and whether that diversity or abundance changes in time, in space, and with relationships to environmental parameters. This example is for the nitrogen cycle, but we can actually do this for other biogeochemical cycles like the carbon cycle, the sulfur cycle, phosphorus, arsenic, and so on down the line. And from the same environment, in fact, the same sample that we collect, we can also collect 16 and 18S ribosomal RNA. So the question is, why do we have to do both? If we know who is there through the 16 and 18S ribosomal RNA, don't we automatically know what they're doing? Well, unfortunately, over the last 30 and 40 years, we've come to realize that a lot of these genes that catalyze transformations are what are called mobile. They're subject to horizontal gene transfer that scrambles connections between this taxonomic gene marker and the functional gene marker. In some cases, from 16S ribosomal RNA, we can get an inkling of what's going on in the system. But if we really want to understand that, we need to collect gene-specific information or functional gene-specific information to really assess what is going on in a system. And ideally, we analyze these in context with one another. So how do we do that? Well, we're an enabling core for Project WiCCED. So we need a process that is going to enable us to handle lots of samples and provide information back in such a way that it's going to be useful to researchers that aren't experts in generating or analyzing this type of data. So we're implementing a method that's already been developed, that's a mouthful to say, a flexible and economical barcoding approach for highly multiplexed amplicon sequencing of diverse target genes. All this translates to is we can measure who and multiple instances of what in one sample for relatively little money, about $30 a sample all told. Now, because we're serving interests of Project WiCCED, we actually went out and we didn't pick what we were interested in. We surveyed, we sent out a survey to the community, and actually we're at a point where we probably need to send this survey out again and see if things have changed, where we asked Project WiCCED researchers, what elemental cycles are you interested in? And then we began collecting information on what ones we could actually interrogate using PCR and amplicon sequencing. And it turns out that what people are interested in is arsenic, phosphorus, carbon, nitrogen transformations, phosphorus, and sulfur, as well as these universal taxonomic markers. Other things that we looked at, all told, didn't have as much interest, so this is what we decided to end up focusing on. So how do we do this and incorporate these particular targets? I'm going to give you a simplified example. So how this works is that from a given microbial community sample, we use PCR to amplify, for example, the NRF gene, which is a nitrite reductase that catalyzes the reduction of nitrite to ammonium in an ecosystem. What that means is that nitrogen is actually retained within the ecosystem and bioavailable to other levels of the food web. From the same sample, we also amplify the 16S ribosomal RNA gene. And it may be difficult to see, but if you look at the PCR primers for these genes, you can see that there's an extra little bit of color that is shared in common between the NRF gene and the 16S ribosomal RNA gene that are coming from this one sample. This is called a barcode tag. From a different sample, we do the same reactions, but we provide them with a different barcode tag. Technically, it's a little bit more complicated than that. We actually apply the barcode tags at a little bit of a later step, but that's not a detail that you need to worry about. Now, in the initial paper that I just talked about, in the proof of concept, they actually tested this using PCR products to represent a fairly simple microbial community, and they tested it on a few environmental samples. But in order to show that this is going to work, we wanted to demonstrate that we could do this really at scale. So what I'm going to tell you about is our first real world samples that target not just 2 genes per sample, but 11 genes per sample, We have one synthetic positive control community. We have one negative control community. And then we analyzed a set of 24 distinct samples that were collected across the state. And these samples were collected and processed in the core by high school, undergraduate, and graduate students using a standardized protocol to see if we can really get very good replication and robust data analysis. So, standardizing the process. Again, using this analogy, perhaps a better and more tasteful analogy, although I find sausage pretty tasty. I grew up in Wisconsin and it's still close enough to Oktoberfest that I still like my bratwurst and potatoes and sauerkraut for meals at this point of the year. What we're doing is we're assembly lining the process of DNA extraction, of sequencing, and of bioinformatics, and then ultimately in collaboration with the data core, we're going to be providing data in an interactive output that's not going to rely on you coming to me and me telling you in a seminar about what we're actually doing. So this may seem like a fairly complicated process, and in truth it is. It took us about a year to make sure that we had it right and that we got it working the way that we were happy with. And so this talk very well could have ended right here if the answer to this question, does this all work, is no, but it turns out that the answer in fact is yes, and I'm going to provide you with that data now. So in terms of does this process work? The answer is yes. So this is data that was from the original proof of concept study. And then we took a look at 3 different ways of analyzing our data. One is a 2 by 150 paired end read, which is short reads of sequencing. A 2 by 300 read, which is the maximum recommended read length for the sequencer that we have access to in the UD sequencing and genotyping core. And then a condition that many people have reported on in literature, which is we're trying to squeeze every last bit of sequencing data out of the money that we're spending on sequencing in this. And what you can see is that when we use an equivalent number of samples and an equivalent sampling strategy and sequencing strategy to what was done in the proof of concept study, we actually get more data out, and we have a similar division of data in between what we call parsed reads, or reads that we can actually analyze, and what's called junk. Although in our case, this junk data, I'm not going to describe it right now, but this junk data, we're actually including other DNA samples that we can pull out and still get useful data from what ends up in this pool. If you have questions about that, I can address that a little bit at the end. So does it work equally well across all samples? So I'm going to show this bar a lot in my talk. This represents the 16 freshwater samples, the 2 sediment samples that we have, and then the soil samples that we've analyzed. Very happily, we have very few sequences that come out of our negative control. That means that we're doing everything in our laboratory very cleanly, and we don't have to worry about subtracting large amounts of sort of contaminating reads from any of our data. We actually get really good yields out of our positive control. And then you see we have a fair bit of variation across the different types of samples and even within a given type of sample. And so we wanted to see whether this variation is really impacting our results, as well as whether the variation in the methods that we're applying actually impacts our results. So we're looking at taxonomic genes, the 16S and the 18S genes, and then this collection of functional genes that correspond to these different elemental cycles. PMO corresponds to a carbon cycle gene. Both the Pho genes correspond to phosphorus cycling genes, nitrogen genes, sulfur, and arsenic genes. I'll come back and talk a little bit about some of the sulfur and arsenic genes, as well as the phosphorus gene later in the talk. What you see here is that our read libraries are dominated when we look at this in a normalized fashion, so by percent of reads, our reads are dominated by 16S and 18S reads, and actually that is an intentional result. We expect that not every organism in our sample is going to have a DSR gene or an MCR gene, so we don't have to sample those genes as deeply in order to get a complete picture of what's present in the community. In contrast, every cellular organism in our samples is going to have either a 16S or an 18S gene, in some cases both. In eukaryotes that have mitochondria or chloroplasts, they have copies of both types of genes. And so we need to sequence much more of these two genes in order to get the information we need to do the community analysis. The other thing that we looked at is that, well, DNA extraction isn't perfect across all of the samples. We end up with samples that don't give us very much DNA. So we asked, did that affect our ability to analyze all of the genes? And the answer is that no. Whether we go with what's considered to be a very small amount of DNA, 0.2 nanograms of DNA in a PCR reaction, versus a standard amount of DNA in the field, 1 nanogram of DNA in a reaction, we essentially get the same recovery of genes across the samples. And there's as much variation within these sample groups as there is between these samples. In general, we get a little bit more DNA out of soil, but that's not terribly surprising because biomass levels tend to be higher in soils than in water samples. A little bit of a technical bit for those of you that are interested in sort of the sequencing aspect. The green here is the data that I've been talking about, the 2x300, or actually believing the company when they say how long you should sequence for on their instrument. The orange here is trying to squeeze every last drop out of the sequencing reaction. And it turns out if you do that, you see a small but statistically significant loss in the total length of sequence that you recover for any given amplicon, irregardless of the length of that amplicon. And 2x150, you get all of the sequence that you paid for, but you end up not having as much sequence in the end. Therefore, we consider that we're going to keep moving forward with a 2x300. And this is one of the interesting findings. Pushing your sequencing to the last drop is actually not good for your data. The other big question, particularly when we're dealing with environmental samples, is this data reproducible? In other words, if we go in and we do the same thing, this is a complicated sample handling pipeline. Is this data reproducible? And this is showing that at a 16S ribosomal RNA phylum level. So what does a phylum actually mean? If you go way back to high school biology, this is Linnaean classification where you've got kingdom, order, phylum, or kingdom, phylum, order, family, class, genus, species. So this is a really aggregated level of taxonomic identification. This is things like animals versus plants versus amoeba in terms of the different groups that we actually can resolve at this level. The nice thing is that qualitatively, all of our freshwater samples have similar patterns of colors across them, meaning that they have similar distributions of these phylum level groupings of bacteria and archaea. Soil samples look like each other, and sediment samples look kind of in between, and they still look more like one another than they do either freshwater or soil samples. So this is a qualitative, sort of estimate that, yeah, this works in that water looks like water, soil looks like soil, and sediment looks like a mixture of the two. But really, this is the key. Quantitatively, if we analyze through our pipeline 2 samples from the same DNA extraction, we get essentially exactly the same distribution of phyla, regardless of whether that sample is from freshwater or whether those samples come from soils. So the two sample sets in blue here are technical replicates of 2 freshwater samples from Silver Lake. These are 3 soil samples sampled from a farm downstate. This is actually called DSS because there's actually a law that enshrines a Delaware state soil. So what we have done by taking these samples is we're using the Delaware state soil to designate the state microbiome of the Delaware state soil. So at some point, maybe our policy folks should get at, putting that into a proclamation. So this works for bacteria and archaea, which I'm a prokaryotic chauvinist at heart. This makes me really happy. Okay, but I will admit that eukaryotes are important. So does it work at 18S ribosomal RNA, which is the marker for algae, fungi, and protists? And the answer is we see a little bit more scatter away from the 1-to-1 line, which is the expectation of if we had perfect reproducibility. But for environmental data, this is crazy good correlation. So this technique and our long, complicated process of barcoding and tagging and tracking, and then computationally ripping this all back apart in the end still works at eukaryotic levels. Freshwater looks like freshwater, soils look like soils, sediments look like a mixture of the two. So this is great. What about the other functional genes? Does it work for them as well? That's where we're going to start talking about some of the vignettes and some of the things that we can actually say about these data and the fact that we used real world samples in our shakedown runs of this process. What can we actually say about what's going on in Delaware? What we're really interested in terms of the science is how do microbiomes and their functional potential vary across Delaware? And then one of the things that I'm really interested in is there microbial novelty in our collective backyard? Do we have sites where we have enriched microbial unicorns just by virtue of the peculiar environmental conditions at that site? And can we then go back to those sites and actually pull those microbial unicorns out and get them into the lab where we can study them and actually learn something about them? Okay, so what can we say? So we have samples that have been collected at nowhere exotic. This is actually collected not even in the middle of the Delaware River. This is just standing on the shore at Battery Park in Newcastle, Delaware. We have both water and soil sediments from there. We've got water samples from Silver Lake. Again, these were collected by Kevin Schumann from Wesley, soon to be Dell State. And then we've had a time series going on with Scott Andres at Coursey Pond through the summer of 2019, where we also took a soil sample. And then the Delaware state soil comes from a farm down in between Milford and Lewis. And we'd really like to thank the farmer for giving us permission to actually go out and sample on this. So what I've done here is at the species level. So the last plots that I showed you were, again, sort of looking at animals, plants, fungi, protists. Here, we're looking at divisions on the order of chimpanzees, bonobos, humans, and orangutans. So much closer divisions and much more fine divisions. We still see that freshwater samples are similar across locations, whether they've been taken from the Delaware River, from Silver Lake, from Coursey Pond. However, we see that within one site, we see differentiation. For example, this blue particular taxon was only present in the first 2 Coursey Pond samples, and it was much smaller in later samples, whereas this gray taxon actually came up. These are all in a time series. And what we're going to be able to do is look at how these taxa change over time at this site. And what Scott Andres is providing us is the environmental chemistry and environmental physical data to interpret what might be driving those changes. The other thing about all of our freshwater samples is that any of the bars that are present and colored with one of these colors, those are cyanobacteria. So cyanobacteria are like Trichodesmium thibautii. They actually fix carbon, they produce oxygen, but they also produce algal blooms and potentially harmful algal blooms because some of these cyanobacteria are potentially toxin producers. And that's actually the focus of a particular project that I'll talk about in just a minute. If you look at our sediment samples, we find limited overlap with the water column and even less overlap with the soil samples. If we take a look at the taxa that are most abundant across all of, most frequently observed across all of our soil samples, what we see is these most frequently encountered taxa make up a smaller proportion of the population in soils, only about 30 to 40%, whereas we get, in some cases, upwards of 75% of the community described by a few species level taxa in freshwater systems. What that says is that soils are more diverse, and that's not an unexpected finding because soils are very heterogeneous. There's lots of microenvironments in soils that are not present in freshwater systems. Again, sediments are kind of in between. They're not particularly closely related to soils. They're not particularly closely related to freshwaters. They represent a mixture of these two types because that's really what they are. The really intriguing thing here is that this blue bar, which is the single most abundant taxon in all of our Delaware State soil samples, is a member of the archaea. Now, if you're thinking about archaea, and even most microbiologists think about archaea as these exotic organisms that live at hydrothermal vents and at hot springs in Yellowstone National Park, this is a garden variety farm field between Lewis and Milton, Delaware. Now we know a little bit about, this is one of those cases where the 16S ribosomal RNA can tell us a little bit about what we expect this organism to do. And these groups of organisms typically are involved in oxidizing ammonium to nitrate and using that as an energy source to provide their growth. Farm soils get fertilized with ammonium-rich substrates. It's not surprising that these archaea would be found in these soils. So it works. This stuff really works, and it's telling us things that we might expect. Now, this is a little bit different of a view, and this is trying to see if we have any universal species that are present absolutely everywhere. So what I did is I looked at the 30 species level taxa that were most frequently observed in all of the samples that we looked at. And what you see is that they tend to still reflect whether the samples were freshwater or whether the samples were soil. A lot of these are cyanobacterial taxa. And what it says is that we don't have one universal inhabitant of these very different environments. So this is actually a good result. The other interesting thing is that roughly somewhere between a third and a half of these taxa, of these microbial species, are not yet cultivated in the lab, and yet they're very abundant. So we have enriched for these microbial unicorns in Delaware. We can go to these sites and try to pull out these species and be able to understand more about what's going on at these different sites and what role microbes are playing. Okay, what about the functional genes, right? So what I've been talking about so far are the taxonomic markers, the 16S and the 18S ribosomal RNA genes that tell us about who is there. And the reason that we've been able to analyze that data further is that the tools and the data sets are more developed to enable us to do that. We're working on the underlying databases for our functional genes, but we can still see some pretty interesting results. So this sample here, CL, corresponds to a sample of soil that was taken adjacent to the Corsi Pond boat landing. So we call it Corsi Landing. And the thing that you notice is that relative to other soil samples, we have less 16S and 18S ribosomal RNA reads, and the bars for the functional genes are much bigger relative to the 16S and 18S ribosomal RNA genes. And this distribution of enlarged categories is not random. What we see is a 6-fold increase in arsenic cycling genes, an 11-fold increase in sulfur cycling genes, and about a 5-fold increase in methane cycling genes. We're digging into this in more detail, but that really suggests that this particular site has probably undergone an anoxic event relatively recently before this was sampled, maybe a flooding event, and it suggests that the distribution of reduced species of species of arsenic and sulfur and methane are probably different at this site than at other sites. We can use this fingerprint as a quick way to assess what sites we need to go study in more depth, and this is definitely one of them since this is a recreationally used site. Now, we were also worried about, I didn't share with you all of the things that we've been worried about, but really we operate on extreme paranoia about whether things are working or not. So we do lots of controls. We try to make sure that things are working. And we had an issue in that in our positive control, we included a species of bacterium that should carry a FOD gene and serve as a positive control for that PCR amplification. And when we did that early on, we said, oh, doesn't look like those primers work. This enzyme is involved in clipping phosphate from organic molecules under alkaline conditions. We also have sort of a partner enzyme that works under acid conditions. So this is really enzymes that are involved in making phosphorus bioavailable to plants. We saw lots of, quote, FOD amplicons in our samples, and we were like, oh, okay, wait, our primers didn't work in our positive control, but they're pulling stuff out of all of our environmental samples. We really need to look at this and say whether or not we're amplifying just sort of garbage or whether or not we're amplifying what we expect. So Alexa extracted for me, because really I'm kind of technically useless except at making some graphs and things. She extracted the 8 most abundant FOD amplicons. What I did is I took the DNA sequence for those, translated that into protein, and then used that to search databases that consist of protein sequences from known organisms, all protein sequences, not just FOD, and looked at the resulting hits that we got back. And I've just arranged these in what's called a phylogenetic tree. Sequences that are more closely together in this tree are more closely related to one another. The nice thing is that every single sequence in this tree is FOD, and our sample sequences that are marked by these little green triangles are actually closely related to FODs, either from isolated bacteria that we expect to have this activity or that have been seen in environmental samples. So we're not amplifying garbage, we're actually amplifying exactly what we intended, even though our positive control didn't work. So it works. And that was the alternate title for this talk, was It Works! Come send us your samples. All right, now briefly in the time that I have left, I want to talk about some undergrad research projects that we're doing. This is Miranda Marini, who I saw was on the call earlier. She's been working now for a couple of years on samples, again, that we just collect right from our backyard in Newcastle Battery Park, and really saying, can we isolate some of these, can we enrich, cultivate, and isolate some of these microbial unicorns in the laboratory? The answer appears to be yes. So if you look at this, this is an earlier analysis that we did where we amplified 16S ribosomal RNA out of cultures that Miranda was able to develop from that site. And we see an uncultured bacterium that is actually very distantly related to known bacterial species. So this would be a new genus. When I talked about species, that is sort of orangutans, that's all the great apes, we're all primates. This would be like a new species of something between the great apes and the lemurs. So this is a really new type of organism. And excitingly, Miranda has colonies on plates from new rounds of cultivation that we're gonna be using our methods to analyze. Now, back to cyanobacteria. This is Alina Brown, who is a former Project WiCCED supported undergraduate, and Miranda has also been generously supported by Project WiCCED. She has been working on the project with Scott Andres to look at Corsi Pond. Corsi Pond early in the summer looks like it's got nice collections of diatoms in it, and in fact, our 18S RNA data suggests that. But in the summer, it looks like you could walk on it because of cyanobacterial bloom. What we're really concerned about here is whether or not these cyanobacteria have the potential to synthesize toxic molecules. It's well known that cyanobacteria make hepatotoxins, neurotoxins, skin toxins, dermatotoxins, and cytotoxins. It turns out that these are all synthesized by enzymes, which are encoded by genes. So we can use the same techniques to analyze this. So what Alina did was develop a custom panel for cyanobacterial toxin genes that we're now beginning to apply using our same methods that we used for other functional genes to assess the time series from 2019 in Corsi Pond. What I can tell you is, unfortunately, I will almost guarantee that we are going to see hits for cyanobacterial biosynthesis genes because the 16S ribosomal RNA is strongly suggestive that there's going to be some toxin producing species in there. Now, Alina is going off to veterinary school, and I'd again like to thank Project WiCCED for supporting us. Alexandra Pepinos is a new undergraduate student who's taking on where Alina has left over and will be actually carrying through the molecular analysis of these time series. So how can we help you, right? So we can analyze samples collected by you by the Delaware Microbiome Project. We can help you with undergraduate research projects that are focused, like those for the cyanobacteria in Corsi Pond or the isolates from Battery Park in Newcastle. Or we can advise and collaborate with you on custom amplicon panels or using our standard nutrient cycling amplicon panel on your research. We're also currently constructing, and this is something that Alexa came up with on her own, a database of functional gene primer sets to help you go through selecting these and help us keep track of what we think is probably a good thing to be able to include in our workflow versus not. And ultimately, as Alexa emphasized, developing these patterns of regional microbial ecology. With that, we have actual kits ready if you want to participate as a citizen scientist or take a sample from a research site that you have. We will happily, for free, provide you with these functional gene sequences and analyzed functional gene and taxonomic gene sequences. This is a picture that I took from our lab where we literally have kits stacked up and ready to go that give you everything you need to give us a sample that we can produce exactly the same type of data for you. We also are running a community sequencing program. Since we're short on time, I want to get to the point about how you can reach us. So it turns out that the website is now live, sites.udel.edu/microbiome, and our email address for the Microbiome Core is also live. It's microbiome@udel.edu, and these will also be linked off of the Project WiCCED website So we really look forward to interacting with you and figuring out how we can actually help you, not just be a government sort of entity that scares you. So with that, I'd like to acknowledge the people that did the work and of course the funding, in particular, Kent Messer, the PI, and then especially Holly Michael, the co-PI, for being patient with us as we really got this up and going and can show that it really is reproducible. And then the people that have done the work. You met Alexa earlier. Alina, I mentioned, along with Miranda. Alwayne Allen was a high school student, also supported by Project WiCCED, who collected some of the samples. And again, the Microbiome Core, we're here to make really good, tasty sausage out of your samples. Thank you very much, and I would happily take any questions at this point.
All right, thanks, Tom. That was great. Really exciting to see what's going on in the Microbiome Core, and you guys are up and running. So yeah, well, actually, we're almost at 12 o'clock. So hopefully some of you can stay and ask questions, but I want to mention before some of you go that our next speaker will be Cal Calvacharla from Delaware State, presenting on December 16th. So hopefully you all can join us then. Okay, so let's take questions, and just either type it in the chat or feel free to just wave at us and we'll call on you.
Okay, so Jeanette popped one up in the text, specifically asking, are there locations in particular that we would like people to sample? And the answer is no. I believe benign neglect, which is allowing people to go where they're interested in. And when you get to the point of submitting a sample, you will be going to a web form and we'll ask you about why you went there. But I like kind of a random walk. Once we get an idea of where we're getting covered, we might actually ourselves go out and hit areas that haven't been sampled. But I want this to really be citizen and researcher driven. What are you interested in? And how can we see how that fits in with the rest of Delaware?
And I will add selfishly, the less places you guys go, the more places I get to go.
No. I need you in the lab extracting DNA and making the robot work nicely. I didn't emphasize it here, but actually, let's see if I can do this. I would have to go back and share the screen, but we actually got a robot out of this grant, which is really cool. I do have video of Alexa monitoring the robot that will be posted, if she gives me permission, on the Microbiome website. So.
Cool. Other questions?
Hello, I have one question.
Great.
Are you hearing me clearly?
Yep.
Good afternoon. So I'm interested in some compounds called polyamines. I'm not sure if you're aware of them, but they're right.
Very well aware of them. Yeah.
Right. So I'm curious to know if there's any protocols, any method that can identify polyamine accumulation in the rhizosphere or the soil surrounding a plant that might be exudating polyamines. And as again, I know they are highly associated with microbes as well, mycorrhizae.
Yeah, so I mean, in terms of the techniques that I just talked about and what the Microbiome Core is able to do for you, we can look into whether there are genes that confer the ability to either synthesize or degrade polyamines, and we can help you analyze the distribution of those genes at various sites of interest. In terms of identifying the compounds, that's not what our core does. The
genes
talk to in that case is actually the sensor core. What they are really tasked with in terms of the research plan in Project WiCCED is interacting with researchers in identifying targets for analysis and for sensor development. So that's, correct me if I'm wrong, I think that's John Raybolt and Wei-Jin Tsai are the ones that are leading that core. So in terms of actually measuring those compounds, those would be the folks to talk to. But what I can tell you in my other hat outside of Project WiCCED is that there are HPLC analyses that are capable of doing this. They're slow, they're cumbersome, but they can be done. So maybe contact me offline if you're really interested in that, and I've got an HPLC that's capable.
All right, thanks for that. But can I, to get a little better understanding, there are techniques that can be used to identify genes that associate with polyamines in the soil.
Yes. Well, what we can do is we can go in and say whether or not a known polyamine biosynthetic gene or a known polyamine degradation gene is present, right? And not only that, not only presence absence, but how diverse is that gene at a given site. So for example, what I expect out of that Corsi Landing pond site that's got that explosion in ARRA sequences is that that's going to be dominated by a very few number of sequences that represent an expansion of a microbial population that's sort of dealing with that. And so it's not only presence absence, it's what does it look inside that box that we're going to be able to help you with.
Very interesting. All right, sounds good. I love that. Thank you very much.
Yep.
So while John had me racking my brains for weird places to go dig up dirt in Delaware, literally, one thing that did occur to me.
We don't discriminate. Dirt would be good. We're a little bit underrepresented on dirt, but sediment we need more of. Yeah.
Well, one thing that occurred to me is simply safety issues. Is that factored in here? I mean, I suppose that rests sort of on the person handling the sample, but I would feel very bad if I, dug up a bunch of creosote from the copper site out back and sent that on to Alexa without an appropriate label saying don't.
So we emphasize in the instructions for the kit that you need to be going to a safe site. For a general Delaware microbiome project, no, and even more, no, please, no bodily fluids, no biological materials. If it looks like poop, it's going in the biohazard. We're not going to identify which dog has been pooping in your yard. Sorry, that's just not what we do. From a research standpoint, we certainly can think about how we might want to do that if there's a project, and that's where I'm interacting with other researchers that may have contaminated sites. This is not a huge deal. The other thing that we are going to find, that I will guarantee you that we're going to find in our freshwater and probably our sediment samples, we'll find pathogens. And, the question is, if we find a site that has a lot of pathogens or a particularly nasty pathogen, what do we do with that information? Who do we talk to? And it's even at the point with this Corsi Landing soil site, I think we really probably need to give DENRIC a heads up and say, hey, I don't know if you guys know about this, but there's probably something wrong here.
Right. I mean, that just came up because, when I start thinking about, interesting things to send, one's mind turns to, oh, heavy metal contamination. That's kind of cool. And so
I think you're at an end of the bell curve there, Chris. Yeah.
Okay. Oh no, that's demonstrable, because the other thing I wanted to ask you is this nice frame print I have over my desk. In the upper corner here is the tidal ditch along 495 with the weird arsenic cycling that somebody turned up about a decade ago, and I don't know which group at UD is doing that, but I hope that's
one of your sites, Holly?
Yeah, we're working on that. It's pretty bad. Yeah.
So, but yeah,
okay.
We've got kits if you're going and sampling, and that would be one that we'd love to do.
Okay.
Yeah.
Good to know. Yeah.
Or Alexa would probably happily go out, or I would happily go out with you and actually collect samples to do that. And there again, since it's a research project, we'd still use the same approach. We'd still use the same primers, but we would be able to collect a few more samples that are a little bit more finely distributed based on what you want to know, based on what you already know about site chemistry or reactivity, things like that.
Yeah, so that site was actually part of our last EPSCoR project.
Yeah.
I think all of our instrumentation is out, but I'm sure we could still get access to the site and then add that to what we know about the biogeochemistry and the hydrology, which would be really interesting.
And one thing to add on is for Chris, you're talking about sampling interesting sites and finding interesting things. Is we're finding interesting things even in the most uninteresting of sites, right? And so even if you can sample what doesn't appear to be an interesting site, there are some really cool things that have come out of that. So one of the examples I have is that, you know, the shoreline for Delaware, right? Where they just stood at Battery Park and sampled some water and some sediment. Within that organism, for example, we found something that researchers previously had only attributed to deep hydrothermal vents. So it's not, yeah, you can find some really interesting things at a very, you know.
Unicorns in your backyard.
Oh yeah, well, I'm not determined to send you something flamboyant, and the really good sites have mostly been capped and remediated anyway. But you know, that's the sort of thing that comes up when I think about this. And yeah, I wanted to make sure I wasn't, you know, sending you a time bomb, as it were, if I decided to sample.
We do things pretty safely, and in the kits, at least from a biological safety standpoint, we have a preservative solution that pretty much effectively knocks everything off. So and also Alexa and myself are trained. We give PPE in the kits. We send gloves along. So we're not sending people out to do silly things, but people are going to do silly things. I'm sure we're going to get kits that we're like, yeah, we're not analyzing that.
Yeah, well, I was mostly concerned about your end. I think there was some chain of thinking here. Battery Park, batteries. Oh yeah, I used to live in New York. Constitution Cove, Hudson Highland. No, Foundry Cove, Hudson Highlands. When you see people in, full sealed PPE because of the worst cadmium contamination in the country. Not that we have anything like that, but it's just like, right, safety. We should think about that.
Yeah, all right. So questions from anybody else that I see on? Hannah, I'm actually curious because I know that we've gone, we've talked around a little bit about an op-ed, and, that kind of got COVIDed. So hopefully in the spring, once the vaccines begin, we can maybe steal a little bit of microbial limelight back from viruses. But please contact me if you've got questions or things that you want to delve into more. I'd also love comments just on the website. It's pretty much bare bones right now, but we will be putting more content and more images up there. The key point right now is to just give people a way to contact us and give us a heads up that they want to see things.
Yeah, I think that's a great idea. I think you had a great point about it being overshadowed a little bit by COVID. So I think that's a hurdle. I'd be happy to reach out. Happy to help also if you need anything with the website. But I'm sure you guys are doing great work with that.
So Tom, I have a question.
Yeah.
So you're going to get a lot of data, and you already, have really good examples of Alexa, interesting things that are coming out of samples that you wouldn't expect. So I guess there are two aspects of this. So, I mean, you're addressing, us broadly in the project and others beyond the project who are not microbiologists, right? And so we don't necessarily know the interesting questions to ask, right? We may have sites that we're sampling and we may know some things about it, but, I don't know what is the cutting edge science in the systems that I'm measuring, but then also on the back end. So I could send you some samples and be like, wow, this would be just interesting to know what's here. But am I designing the sampling in a way that's going to get at the interesting questions? But then on the back end, let's say I would get data, then I don't have the context and the expertise to put this into a paper the way it needs to be put together. And so once you start to get a lot of data, how are you going to manage both the front end of, trying to help people design what they're doing, and the back end of, I imagine that you're going to be involved heavily in the science that comes out of it, right?
Well, right. And so again, this is not an unusual, this is a really common problem in all of microbiome science. Is you get sort of distracted by the low-hanging fruit and you go, oh, look at that, something shiny over here, something. And so the question is, what do we do? That's where we have this goal of standardizing. And that's the other thing, is that, I'm sorry, this is going to sound like blasphemy to an environmental scientist, but I really want things to be sampled as absolutely similarly as possible so that we can intercompare data, right? If we don't have reproducibility, and that's why we put together this kit, right? Where we know that your sample is going to be of a defined size, which means we're going to get some information about biomass out of that. It's going to be put into, within a reasonable amount of time, a stop solution that's going to preserve it, so we don't have to worry about sample changes during that. So we've already tried to, on the front end, address that by trying to make this process as, just here's what you're going to do. And really, it's just a matter of, it really is as simple as you knowing that you have a site where, let's say, a reaction is going very fast. It's, in a 1 meter zone that's 4 meters below the beach. Get us that sample. We'll tell you microbially what's going on there. And,
okay, go ahead.
Yeah. So the point is that because we're doing that sampling the same way that we're doing it for everything else, we actually have global context for your sample, not just site context for your sample. And
right.
By virtue, and all you have to tell us is what, if you want to do something that is beyond what we have in our standard panel, that's where you talk to us, and that's where the database that Alexa is working on. There's actually fascinating stuff there that I didn't talk about, where she's talking about using natural language processing to go out and identify in the literature what primer sets are for different processes, things like that. So we can expand our catalog of things that we can offer. So we can talk with you, and in a relatively short span, be able to say, okay, you're interested in this. If your interest is really only on arsenic and phosphorus, and the carbon doesn't matter so much, we can generate a custom amplicon panel that we would process the samples that are still done the same, that are still collected the same, but we would just analyze for different targets. I know it's suspiciously easy, but that's what we're trying to do. And then let me address the back end.
Yeah.
And quickly on that. So yes, we totally understand that you're not experts in sort of the data analysis in this. And again, that's where we kind of liken ourselves to the FDA. We are trying to provide you with a standardized product that meets the accepted standards in the field for what good data is. And then we help you interpret that data, at least at a first pass. And then we can help you, and ideally from a long-term sustainability standpoint, collaborate with you moving on out in the future for sort of subsequent projects about how do we take this first pass data that indicates these interesting things, and how do we capitalize on that? That's what an enabling core to me is all about.
Yeah, well, that's what I'm asking, because, right, we need the expertise in addition to Yeah, right. So even if I get the best data, how do I know what to do with it?
Well, no, but that's where with the selection of genes that we target up front, we Alexa and myself look in the literature. You say you're interested in arsenic. We look in the literature and we see what we can do and what that can tell you about your system, about linking genes to transformations. Mm-hmm. And then we can say, oh, it looks like we can point to a bar in the sheet that we give you and say, look, this arsenic reductase is way less diverse than it used to be, and it's way more abundant than it used to be. So yeah, right there, that is, and we can provide the statistics on that. And again, we're doing, because we are standardized, right? We've got a really good handle on what those acceptable standards are, right?
Yeah.
In some ways, and again, it's a little bit blasphemy, but irrespective of the niche that you happen to be in. The other point is that this type of connection between the microbiome data and the flux data and the biogeochemistry data, it's still pretty early days on that. So anything that you get in this,
that's true.
Pretty good.
Yeah, that's true. Yeah, great, thanks. Anything else before we wrap up?
Just for Kevin, 20 kits? No. we don't have 20 right now. We can give
That's okay.
We can give you 6 for right now, but
that's fine. That's fine. I was starting to think, 20 may be a little too many.
20 was a little greedy. I can give Kevin a little stick because he's my former grad student. So
no, just let me know if there are any other sites in the Silver Lake area or anywhere along the St. John's you want sampled.
No, I think a time series of those same sites annually for the life of the project would be really good. Even seasonally would actually be really good to go back to the same place in seasons, and then we construct time courses for this.
Yeah.
Holly, some of it is really, again, it's a little bit more of a discovery based rather than hypothesis driven, but I've really come to understand, and well, I can't believe NSF funded it, but I'm going to run with it. If they're going to let us do discovery-based science, by God, I'm going to do discovery-based science.
But in theory, like you said, the discovery helps us develop hypotheses.
Exactly. It helps us tell us about who the likely important players are. And the other thing you can do is that when we do the functional gene, you get other taxonomic information from the functional gene. So you can sort of say, hey, is there a match between the 16S and the functional gene data? Or is it that you've got a small population here that's working really hard to catalyze a particular reaction, right? And even that type of insight about the abundance of your catalyst within the larger microbial pool is still valuable to have, and actually, or at least relatively quantitative.
Yeah. So I'm actually really interested in that kind of thing, looking across salinity interfaces and seeing how.
Yeah, so the problem is there's not a magic gene for salinity, but the fact that we saw what we did at the Corsi landing, I think we will have no problem at getting a fingerprint that's community-wide, right? That we will actually be able to. So again, if you've got sites, we do these by standard methods, I think we'll get to a salinity fingerprint. And that fingerprint is going to linger beyond the transient intrusion and walk back.
Yeah, which is interesting. The other thing that's interesting, though, I think, is, if you have area like in the subsurface, zones that are always fresh, zones that are always saline, and zones that are always brackish or really oscillate, I imagine that you end up with a very different selection, like you said.
Oh, yeah.
It gets smaller versus big. And I think that would be really interesting because it would.
Yep. No, and yeah, we can. That's the type of thing that we can do that, and that's again where standardization and not being sort of boutique-y about how you do the microbiome stuff. That's where it actually helps you, because you can say, look, if we did something wrong, we did it wrong completely consistently.
Yeah.
And if there is any bias, which so far, knock on wood, we really don't see much of a bias. We do see a bias in the 18S primers, but we already knew that was going to happen because other people had complained about it. It turns out that the 18S gives you a lot of bacteria that I had to chuck out of this. And it's not very good for eukaryotes, but we've figured out that we can include another one that'll do fungi a lot better, and that gives us good coverage. So.
Cool. All right, well, I think we should probably wrap up unless there's anything else. But that was great. Thank you, Tom, and thanks everybody for coming.
Thanks so much for having me. And yeah, happy retirement.
Thanks, you too.
All right, talk to you later.
Bye.